So like I said, welcome to Genomics Up. We are a purpose-driven startup company building maximum impact in the biotech space, and we understand that there's quite a lot that needs to be covered as far as is concerned. So, which is why we're coming up with this program, which is exploring comprehensive genome analysis. All right, NGS data analysis to be precise. So, excuse me. All right, amazing. So this is comparative genomics and its application to microbial plants and animal research. I repeat myself, the topic we're going to be treating today, the aspect of genomics that we're going to be covering is comparative genomics and its applications to microbial plants and animal research. All right. So what is genomics? I know you must have been familiar about genomics or what genomics is all about. And I know probably some of you, this is the first time you've been knowing much about genomics, or this is the first time you're hearing about bioinformatics, all right? So this is a field of biology that focuses on the study of the complete genetic information of an organism. Now that is genomics, all right? That is genomics. I was telling a certain group of people earlier today that there's difference between genomics and bioinformatics. I repeat myself. There's difference between genomics and bioinformatics. Genomics is an aspect of genetics that is focused on the DNA sequence of an organism. And in this DNA sequence, this is where you explore the genes, the genome, and some certain functional proteins, including hypothetical proteins, like proteins that does not have any function. That's hypothetical protein in the genome of an organism. So that is genomics. And genomics could either be at the genome level or it could be at the gene level. All right? It could be at the genome level or it could be at the gene level. Please tell Tosin to join the Microsoft Teams. So if she can hear me, she can indicate that nobody can hear me. Thank you. All right, so sorry for that distraction. So that is genomics for you. You can explore genomics at gene level, or you can explore genomics at the genome level. Now, when I talk about gene level, I'm talking about a certain DNA sequence that is coding for a particular protein. Now, but when I talk about genomics, I'm talking about the entire genetic structure of an organism. Now, this is genomics. So because you cannot explore comparative genomics adequately, all right, you cannot explore comparative genomics accurately or adequately without actually exploring genomics. Do you understand what I'm saying? So, and that is why we have to explore this. So we have to define genomics so that you can understand what we are about to explore. So, and we say that genomics includes the analysis of the structure, the function, and the evolution of all the genes and interaction within organism's DNA. I repeat myself. This includes analysis of what? The analysis of the structure, function, and evolution of all the genes and interactions within an organism's what? Genome. All right, let's proceed. So there's what we call the DNA and the flow of information. I repeat myself, there's something we call the DNA and the flow of information. And you need to understand what DNA is in as much as this particular workshop is concerned, because we need to explore the theoretical part before we dive into the hands-on practical session. Because this is what we give meaning. This is what we give meaning to. To understanding the practical applications of microbial genomics in either plant, animal, or microbial. Now, we started a 4-month webinar series like 3 months ago. That was like February, all right? And we last month we started this comparative genomics, but we only explore an aspect of the hands-on practical session. So we promised everybody that we're going to explore this aspect of this aspect of research, this aspect of genomics in detail, and then we are going to do this for the next 2 days, including today and tomorrow. So then last time also I explained DNA and the flow of information. Now it will be would actually be exploring this area, right? From this end, and I would like to explain this in detail, in kind of more detail, so that you'll be able to understand what we are actually talking about. All right. So DNA and the flow of information. We understand that DNA is the genetic materials that encode the information necessary for an organism to develop and function. I repeat myself. We understand that DNA is the genetic material that encodes the information necessary for an organism to develop and function. And then we understand that the entire genetic makeup of an organism is actually encoded by DNA. Do you understand what I'm saying? Be it the structure, the function, the morphological, the physiological, either plants, animal, or microorganisms. All these things that I just said are actually encoded by DNA. So it means life forms emerge from the DNA structure because they are the genetic material that encodes hereditary information, all right, of the organism to develop, to function, to carry out functional protocols. Now, what is DNA? DNA is what we call double-stranded, all right, ribonucleic acid, okay? So, and then this double-stranded is in a complementary strand. So, but there are 4 alphabet that encodes the DNA. That is the A, the T, the C, and the G. And that is the A, the T, the C, and the G. So, and this is describe the basic molecular dogma where we begin to talk about the purines and the pyrimidines. All right, the purines and the pyrimidines. And we understand that there's double bond between A and T. There's double bond between A and T, and there's triple bond between C and G, please. All right, there's triple bond in C and G. So we get to understand that the C and G are as triple bond, and hence it makes it more stable than A and T. In genomics or in bioinformatics analysis or in genomic study, there's something called GC content. Now, this is the content of G and C in the entire gene structure or the genomic structure. Do you understand now? So that is what we call what? The double-stranded, DNA structure, okay? The A, the T, the C, and the G. Now, before I proceed, I would like some you guys to give me an answer at the chat room. Very quickly, what is the full meaning of A, the T, the C, and the G? What is the full meaning of the A, the T, the C, and the G? Very quickly. Very quickly. Somebody should give me an answer at the chat room. What is A? What is the full name of the A, the C, the G, and the T in the DNA structure? All right, from the Google Meet, we have okay, adenine. All right, we have the thymine. All right, okay, I'm beginning to see the results from the Microsoft Teams as well. Amazing guys, amazing guys, amazing guys. All right, cool. So somebody, okay, amazing. So thank you guys. It means that you guys know what I'm actually talking about. All right, it shows that you know what we're actually talking about. So now this 4 alphabet encodes DNA, which is the basic hereditary material. So we have the DNA, which transcribed to RNA. All right, we have what we call the DNA, which was transcribed to RNA, and we have RNA, which transcribed to, which translate to protein. So this is what we call transcriptions and translation. So if a particular gene is encoding a functionality in a particular organism, the DNA actually encodes, all right, this functionality. But there is a flow of information. There's a progress. And this progress is that it starts from the DNA down to the RNA, and it is so when DNA is transcribed to RNA, that's what we call transcription. But then this mRNA is still at RNA, all right. So, but when we have it as mRNA, then splicing takes place, all right. Splicing takes place. Splicing takes place, and if splicing takes place, it means it is a process of removing the introns from the whole genome sequence, or from the old genetic sequence. All right, do you understand the DNA sequence? So now so the full transcription is now where we have the mRNA when the introns has been taken away. Then we now have the protein, which is the translation, where the mRNA are coded actually for that protein. Now this protein could be either structural or functional. So it means a protein could be functional when it's coding for a particular function in the organism, and it could be structural when it's coding for a particular structure in the organism. I hope that is very clear. So can you reply the chat room? I hope that is very, very clear, guys. Amazing, amazing. Thank you for the answer, because I really love my class to be very interactive, because so that we can be on the same page, right? So that we can be on the same page. All right, let's proceed. We have what we call codons and translations. We have what we call codons and translation. So codons are actually 3 nucleotide bases that encodes, that code for a codon, right? That made up a codon. 3 nucleotide bases. So if we have a genetic sequence or DNA sequence, now out of this DNA sequence, 3 each, progressively, right? 3 each progressively in the DNA sequence made up a codon that codes an amino acid, and that amino acid is what we call the protein. All right? So now you may be wondering what a DNA sequence is. Some of you may have known a DNA sequence, or you may have seen a DNA sequence. Now, just to break it down for you, let me show you an example of a DNA sequence. Now, what I'm showing you right now is what we call Genius software, which is one of the tools we're going to be sending. And actually, we're going to send it in the next few minutes. All right, we're going to send it in the next few minutes. So, and I'm going to be showing you DNA sequence, what DNA sequence is all about, and how this DNA sequence encodes the A, the C, the T, and the G. All right. So now this is what. So this should be a DNA sequence, fragmented sequence of a particular DNA or of a particular gene in wall. How many of you know wall fish? All right, wall is actually wall should be a fish, right? Right. So this is the DNA sequence of a particular gene in wall. So we have other ones. So, but I'm going to show you a practical process where we're going to be going through DNA data retrieval from online tools, such that you'll be able to do that on your own when you are free. All right. So this wall, and in the wall, as you can see here, we have DNA sequences. So this is Genius software. We're going to show you how to use Genius software, because this is one of the softwares we're going to be using. And then we're going to send you a link on WhatsApp group, guys, on how you can actually download this DNA sequence. All right. So, but let me just try to unwrap this. So this is, don't worry, we're going to explore this. We're going to explore this. So now this is DNA sequence. Let me change the color so that you understand what I'm trying to say. Now, can you see this is DNA sequence? Let me try to maximize. So this is DNA sequence, and let me try to zoom up. So this is DNA sequence, and as you can see, we have the A, the T, the G, G, C, C. So this should be a DNA sequence of a particular gene in a wall of fish, like organism, the genome of organism of wall as a fish, right? So, and this is the DNA sequence of a particular gene, unknown actually, though. We don't know what it is, except if we blast it. Okay, you guys are unable to see my screen. Really? Thank you guys for the notice. Can you guys still see my screen right now? At Microsoft? Can you see it? Amazing, 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 amazing. Thank you guys. So let's proceed. Let's proceed. Okay. So let me maximize this. So you should still see my screen. Let me know if you can hear me, if you can't hear me as well, so that we can solve that before we proceed. All right, so I was talking about DNA sequences, and I was trying to say that DNA sequences actually is encoding ACT that made up a gene or a genome. Now, this is just an example, the A, the T, the C. So this is the genus sequence of a DNA sequence of a particular gene in wall. So now, there's something called translation, all right? So because we're explaining codon, remember? So now I'm going to translate this particular DNA sequence using this particular tool on this software. So I'm clicking translation. Now, as you can see, we have common colors, like 4 colors that made red. So wherever you see red is adenine, T is thymine, G is guanine, and C is cytosine. Now, wherever you see this one, you could see that this is an amino acid. Why? Because we can see 3 codons. You get my point? We can see 3 codons, which is the A, the T, and the G. I repeat myself, the A, the T, and the G. The adenine, thymine, and guanine. Now, at this point, we have translated it, and you can see that these 3 codons is encoding this particular amino acid. So this is what we call codon, 3 bases, and coding for this particular amino acid. And this amino acid, just look at this end at the bottom of my mouse. I'm taking my mouse back to the amino acid right now, but while I'm taking it back here, kindly look at the bottom so that you can see the amino acid I'm talking about. And I've put my cursor on M, and you can see that ATG code for M, and this M is an amino acid that is called what? Methionine. So this is the basic molecular dogma. This is how genetic information is being encoded in the genome of an organism, or in the gene of an organism. If a gene is coding for a particular function in the body, it is because there are DNA sequences that made up a codon that is coding for amino acid that is being aggregated together to code for the particular function of the gene. You get my point? For that particular function of the gene. So that is what I'm trying to say. I'm trying to talk about codons and what? And translation. Now remember, even in translation, we have the stop codon. You get the point? We have the stop codon as well. So these are the codons, UUU and UUC coding for phenylalanine, UUA, UUG, leucine, and the likes. Now don't forget there's difference between DNA and RNA. And what's the difference between DNA and RNA? I want to know very quickly. At the chat room, what's the difference between DNA and RNA? What is the difference between DNA and RNA? Very, very quickly. I need the reply. What is the difference between DNA and RNA? Can I see the Google Teams? Google Meet rather. Okay, single-stranded thymine is exactly. So thymine is changed to what? To uracil. All right, thymine is changed to uracil. I hope you guys can hear me very clearly. Can you hear me clearly? Don't worry, somebody's raising his hand. We're going to answer questions in time. So let's proceed. Let's proceed. Hope you can still see my screen. Yeah. All right. So genomics research is aimed at, now we're diving into genomics research, DNA, RNA sequencing, where we talked about whole genome or targeted sequences. Now, whole genome is like NGS data analysis that covers the entire genomic structure of the organism. Then we have targeted sequences, which is specifically talking about a particular gene or DNA, all right, a particular gene rather, in the whole genome of the organism. Then we have analyzing the structure and function of the DNA. So genomics research involves analyzing the structure and function of the what? Of the DNA. Then we have identifying the genes and their functions. Please give me white cup, the glass cup. Thank you. So we have identifying the genes and their functions. So genomics research involves what? Identifying the genes and the functions of an organism. Then we have studying gene expression and regulation. That's also part of genomic study or research. Then we have analyzing the genetic variations and mutations that take place in an organism. Now, each of these has a very, very, very specific, very specific applications in genomic research, because we cannot actually adequately explore and understand a disease or understand a disease in human, a disease in plants, a disease in animals, or even the genetic makeup or the functionality of a particular bacteria, except you do DNA sequencing. So DNA sequencing is the process of getting the DNA, like extracting the DNA and sequencing through PCR in order to get the whole genome sequence or whole genetic makeup of a particular gene from a particular organism. Now we have the analyzing of the structure and function, because before you can actually know the structure and function of a particular DNA, you must be able to analyze it. So it means you have to analyze them. You have to analyze the DNA to be able to know what they are encoded and what a particular DNA structure or feature is found in the DNA. Then we have identifying the genes and their function. So you must be able to identify the genes and their functions to be able to adequately carry out your genomic research. So, and then genetic variations, probably in clinical genomics. So we are analyzing the genetic variations and mutations, and this particular aspect is actually applicable in humans, animals, or plants, where you begin to understand, explore the, in microbia, probably to track surveillance, like outbreak in a particular region or pandemic. Then genetic variations is applicable when you want to track down, a particular disease of a person, or you want to apply it. It's applicable in precision medicine and the likes. Genetic variations, or you want to understand evolution in an organism. So understanding the genetic basis of traits and disease across species and developing new treatment and therapies based on this knowledge. Understanding the genetic basis of traits and disease across species and developing new treatments and therapies based on this what? On this knowledge. So this is an aspect of what genomics study. I'll repeat myself. This is an aspect of what genomics study. Please give me a minute. I told you to turn the fan. All right, so sorry for that little break. We have to adjust something here. Okay, so let's proceed. So when you explore genomics research or comparative genomics, you'll be able to understand genetic basis of traits and disease across species and developing new treatment and therapy based on this knowledge. Now, this is an aspect of comparative genomics because you cannot accurately identify a specific unique trait in an organism without comparative genomics. So it means that whatever area of genomics or bioinformatics you actually carry out, you cannot do without comparison. Are you getting my point? You cannot do without comparison. If you are a life scientist, you're interested in genomics, you're interested in bioinformatics, it is very, very, very important that you have a basic background, fundamental background in comparative genomics, because this will really help you to be able to adequately come up with insights, informations that are needed in order to tackle a specific problem or come up with a new discovery and innovation in the life science space, and disease across species and developing new treatments and therapy based on this knowledge. Okay, so this is an aspect or practical applications of comparative genomics in genomics research. Okay, so let's proceed. So what is comparative genomics? This is a field of biological research in which the genome sequences of different organisms across different species. Don't, it's too loud, okay? Across different species are compared, okay? This is a field of biological research in which the genome sequence of different organisms are compared. It could be a comparison between the same or different species or the same species but different strains, as in the case of microbial organisms. I hope that is very clear, right? It's a field of biological research in which the genome sequence of different organisms are compared. It is from this comparison you now begin to come up with insight, traits, difference in functionality, difference in structure, all right, at the genomic structure level, and the likes. So let's proceed because of our time. Now, this is the research applications of comparative genomics. Research applications of comparative genomics. The first research application of comparative genomics is microbes. All right, microbes. Now, if you're talking about microbes, we are talking about the microbial world, where we talk about bacteria, we talk about fungi, and the likes. Then we have comparative genomics. It's used to understand the evolution and diversity of bacteria and viral pathogens that cause disease in humans. This particular comparative genomics could be used to track down outbreak in a particular area. It could use to track down a specific gene that has been breaking out, that has been, mutating, that has been, because in comparative genomics there are other specific areas of genomics as well. So that is why we're talking about a particular genomics research where we're talking about variants. So if you're exploring comparative genomics, mutations is also part of it, which is SMP, single nucleotide polymorphism. That's comparative genomics for you. Because for evolutions to emerge, there must have been mutations, there must have been effects. So it means that you can use comparative genomics to study environmental effects on a particular microbe. Environmental effect on a particular gene of human. You can use it to comparative genomics to study toxic effects, spillage in a particular environment. So comparative genomics is used to understand the evolution and diversity of bacteria and viral pathogens that cause these diseases in humans. This information can be used to develop new treatments and vaccines to combat this pathogen. The information you develop on the comparison, because it will give you a kind of a data-driven insight. Data-driven insight by the time you carry out comparative genomics. Don't worry, this is just practical to build your knowledge. We are going to explore the hands-on practical sections, because we have today and tomorrow, but you need to be built, you need to have deep understanding such that you'll be able to use this to apply to your field of study, your research, probably master's, PhD, undergraduate, or whatsoever. You want to come up with a research paper, or you want to apply this to your CV, to your research, or you want to use this in your workspace. So comparative genomics is used to study the evolution and ecology of what microbial communities in different environments, such as in soil, in water, and human gut. An aspect of comparative genomics is what we call metagenomics, where you understand the diversity, like the basic abundance of microorganisms in this environment. You get my point? Do you get the point now? So that is it basically. That is it basically. That is it basically. Evolution. To understand, how this community interact with each other. This can help also understand how microbes interact with each other and the environment, and how they contribute to important processes like nutrition cycle and carbon fixation. So it means you can use comparative genomics to study the effect of microbial organisms in agriculture. I tell you, in plants. So it means you can link, you can do a kind of multifaceted research with comparative genomics, where you combine data of microorganisms and that of plants. You combine it, a kind of multifaceted dimensions of research. That is comparative genomics for you as far as microbial organisms is concerned. Because by the time you begin to explore these microbes, you get to understand that they are specific genes unique to the reference sample, or to the reference genome, or to that particular bacteria that has emerged as a result of evolution, emerged a specific virulence or toxic gene as a result of evolution. So let's proceed. Now in plants, comparative genomics is used to study the evolution of plant genomes and the mechanisms of plant adaptation to different environments. I'll repeat myself. Comparative genomics is used to study the evolution of plant genomes and the mechanisms of plant adaptation to different environments. This information can be used to develop new crops that are more resistant to pests, diseases, and environmental stress. So by the time you begin to explore comparative genomics of a whole genome of a plant, guess what? Guess what? By the time you now begin to look at the entire genomic structure in comparison with another genomic structure of a plant, then you now begin to discover that, yeah, there's a specific conserved region a specific conserved region that is unique to that particular genome. And then when you begin to study that conserved region, you could be a new gene as a result of mutations, probably through frame shift on the genomic structure of the organism. You get the point? Or it could be as a result of new mutations. And as such, new genes comes up or mutated genes comes up. And that is why strains are strains, and we keep having new strains of microorganisms because of evolution, mutations that are occurring. But how you know the uniqueness of these things is through comparative genomics. That's it basically. So comparative genomics is also used to study the genetic basis of plant traits that are important for agriculture. Now, by the time you use comparative genomics, you discover that, oh, there's a specific trait. For instance, maybe you have a plant and they are the same species, but there's different subtypes. And I begin to understand that, oh, this particular species of a plant takes 6 months to bear fruit. But there's this one that accidentally, it began to bear fruit in 3 months, probably because of environmental factors. But you can actually do comparative genomics, study the genome of the plant, first plant, the genome of the second plant, to know these traits, to identify that particular new conserved region that has emerged in the new plant that makes it to be yielding 3 months earlier than the speculated time. I hope that is clear. Please, I need an answer in the chat room. Is this really, really clear? I need an answer in the chat room. Okay, so they say it's time. It's Rosa time. They need to take a break. All right, so you guys can actually take a break. Don't worry, you can meet us. So since the practical sessions is available, so you guys can actually get access to it, okay? So you can cover up. So you guys just still be online, do your prayer. So once you are done, you join us back, all right? Once you're done, you join us back. So whichever part you missed, you get access to the recorded session so that you can cover up. So still be with us, stay with us. Just pray your prayer and then get back to us, okay? Is that clear? I need a chat. I need a reply. I need a reply very quickly with a yes, if you understand what I'm saying. Yeah, very clear. Thank you, guys. Thank you, guys. I hope you guys are enjoying this. Are you guys getting this? Are you enjoying it? It's interesting. If it is, you can reply with interesting. Interesting. I need a reply, interesting, before we proceed. Amazing. Thank you. Abu Bakari. All right. Thank you, at Haina. Amazing. So it shows you guys are really following. Shows you guys are following. It's amazing, guys. All right. So let's proceed. So that is it. So by the time you now get this particular new trait in that particular plant, you can now study that particular trait and then see how you can actually modify these traits to make the 6 month one. So that means you have come up with a huge discovery, like with a novel discovery that can actually help a lot in other kind of research and in the future research and in modifying the plants and the likes. Okay. So let's proceed to animals now. All right, so in animals, we have comparative genomics, which is used to study the genetic basis of complex traits in animals, such as behavior, disease resistance, adaptation to different environments. This information can be used to develop new treatment therapies for animal health and to improve breeding programs for livestock. You get a point? So it means you can use comparative genomics to study the genetic basis of complex traits in animals. When you now begin to study, oh, there's this particular trait in this animal that is different from other traits. Probably an animal is kind of a bit intelligent than the other. If an animal is kind of more intelligent than the other, like a dog, a particular dog is more intelligent than the other, you could actually study the complex traits in this animal by comparing with those that are very, very dull. You get a point? And then you now begin to understand, okay, this is the conserved region, particular region that is kind of associated to the intelligence traits in this particular animal. It could be the color. It could be the color. So these traits could be some of them could be dormant, some of them could be recessive. You get a point? Some of them could be disease resistance, probably because of the immune adaptation or immune response, right, of that particular animal, an adaptation to different environments. And then you could begin to study this trait and see how these traits could actually be engineered, or further research could actually be done in order to come up with more improved traits or breed of animal in the livestock space. All right, so we have the comparative genomics also used to study the evolution of animal genomes and the relationship between different species. This can help us understand how animals adapt to different environments and how they have evolved new traits and behavior over time. Because it's true. It's true. You can, okay, for instance, you guys can understand. I really need a reply. Like I need a reaction with what I'm about to say. Now you get to discover that we have seen levels of civilization. You get the point now. Now, can you just imagine the level of civilization we have experienced in our time in 21st century? Compare it with 20th century. What do you think? Man, look at AI, the recent development, the recent trend in the industry, which is AI. I need you guys to interact with me on this matter. Like as humans, right? As humans, we've evolved. That's the truth. We've evolved. We've come up with new traits of intelligence. We've come up with new innovations, with new developments. It is because of evolution. It's because of our involvement. I need you guys to interact with this, sincerely speaking. I need comments. What do you think you can say about what I'm talking about, such that you'll be able to understand the huge importance of comparative genomics? It means that comparative genomics could actually be applied to any industry, even including the AI, in the sense that, oh, if somebody come up with a new innovation, you can actually study this human and understand, guy, what's the adaptation? What's your complexity? What's the trait that is surrounding this person? Yeah, ChatGPT and the AI evolution, and human beings came up with this. So it means that these guys must have been intelligent. So it means that if you have a human being that is dull and a human being that is intelligent, you can actually study these two people to understand, come on, what is the trait behind these guys? And then you could come up with a kind of innovative discovery, like a kind of so that is DNA for you. That's science for you guys. That's science for you. You get the point? So that is it. I'm enjoying this interaction, really. So let's proceed. So research applications of what? Of comparative genomics. Research applications of comparative genomics. Research applications of comparative genomics. Now, we'll be looking at the hands-on practical sessions. I have a workflow with us where we'll be working with today and between today and tomorrow. All right, between today and tomorrow. So we'll be looking at the applications of this comparative genomics, like hands-on practical applications in some of this field. All right, in these areas, and then we've discussed the research applications as well. That is covering the essence of the practical applications of comparative genomics in research, and how it can really help by identifying unique genes, unique regions, conserved regions, mutations that you cannot do any bioinformatics, and even in transcriptomics, gene expression analysis, variant analysis, clinical genomics, pathogenomics, metagenomics, epigenomics, guys. You cannot do without comparative genomics, right? So in the period of this 2 months, because comparative genomics is wide, but we're going to be covering some certain areas which involve data retrieval from the database, which is NCBI in particular. We'll be looking at some software tools for comparative genomics, and then we'll be looking at general properties table, like based on research paper concept, how you can come up with general properties table of different data sets. And then we have genome assembly and genome annotation comparison with genome software. All right. And then we have bacterial genome annotation for real data. All right. Then we have last alignment for conserved regions and unique genes. We have pangenome and evolutionary study. All right. So this is the workflow for the hands-on practical training that we're going to be doing on NGS data samples. All right, on NGS data samples. All right. So I hope it's clear. I need a reply. I hope it's clear. The workflow is clear, guys. What's that? All right, let's proceed. So the first thing we're going to be looking at is data retrieval from the database. So we've talked about DNA, we'll talk about DNA sequences, and then we've talked about NGS data analysis like whole genome sequence, and then we talk about targeted gene like specific genes. All right, specific genes. So let's cover this. So let's start with data retrieval from the database, which is NCBI database. All right, NCBI database. So we need to collect genomics data from this database. So this is the first practical session. So we've already started practical sessions already. All right. So hands-on practical start already. So but before before I go through the hands-on practical sessions, I would like to take questions. I would like to take questions from the previous presentation. Guys, send your questions at the chat room. Send your questions at the chat room, okay? So I will take questions before we start the practical session. So hands-on practical session, we start today and then end tomorrow. So we're going to send you a link to download the Genomics software. I'm going to send you a link to download the Genomics software. So I'm working on it. I want to send you the link to download the Genomics software. So I'm sending the link to the group. The link will direct you to LinkedIn. It will direct you to LinkedIn, and on that LinkedIn, you'll be able to get access to. You'll be able to get access to. So make sure you follow us on LinkedIn. But all the details you need to download the software is on LinkedIn. This software can only work on laptops. So those of you that currently have a laptop, so you could actually post this. You could actually work with this right now. So just go through to the LinkedIn, download the software that works best for you. I'm waiting for questions, guys. I'm waiting for questions. Just keep sending your questions from the previous session that we just had right now. Genius software is not free. It's paid software, but you can use the trial version. So we could have a package for you by tomorrow because we have the lifetime activation key. But first, work with the trial version because I think the trial version is for like 2 weeks or a month. So work with the trial version first before we proceed, before we give you the package. All right. So the Genius software itself costs like $200 per year for students, only students. $200 per year for students. So, but we have the lifetime access activation key. But we have a package for that. By tomorrow, we'll let you know whatever that is for you to get access to the activation key. But for now, please use the trial version, okay, to be able to do this. So because I'm going to take you through how to run, how to work with the Genius software as well, okay? I'm going to take you on how to work with the Genius software. So we have a question. I believe you guys can see the question also on the screen. So how can we apply comparative genomics between two organisms or different species? Amazing. I love this. I love this question. Yeah. So how do you apply comparative genomics between two organisms of different species? Now, if they are different species, they could be the same genus. Do you understand? They could be the same genus. Now, there's something we call copy number of genes. Copy number of genes. Now, this copy number of genes are genes that are present in a species and is present in another kind of species because Generally, bacteria, they have certain common genes that exist between them. All right, common genes that exist in them. So even if you work with E. coli, you work with Staphylococcus aureus, you work with any kind of microbial organisms, you still find some genes that is present. So it's just that the mutations could be different. All right, the differences could be at the genetic level. But when you now begin to compare two organisms of different strains, you now begin to discover that there are certain genes that is present in a species and it's not present in another species. Do you understand me? That is not present in another species. Now, if you get a particular gene of a particular species from a particular species that is novel, there could be a way you can engineer that gene, because you want to improve other kind of species of the same genus. Now, the reason why you could do that is because they are related, and at such a gene could actually be modified to fit into the other one. Another aspect of comparing two organisms of different species together could be that, when you compare these organisms and then you now begin to predict certain variants or mutations between them, and then you could discover that a particular gene could carry a genetic biomarker that could be used to treat a disease or to deal with a particular disease from the other kind of species. So these things ranges, they cut across different stuff, different research areas and applications when it comes to comparing two organisms of different species. For example, you can compare two organisms of different species at the phylogenetic level. Why? Because you want to see if they are genetically related. How closely related are they? And you could do this in drug design and discovery process, probably when you want to study, you are looking for a animal trial, and then you're looking for a specific animal model that you want to use for the trial. You could actually do phylogenetic analysis just to understand the closely related of these animal models in order to see, which one is actually closely related to the alternative, to the main one you have, and that could be the alternative animal model for your clinical trial. I hope that is clear, guys. I hope that is clear. So when you talk about comparative genomics, you often mention conservation. Does it mean one would need to compare a population of which group I am comparing? Yeah, you can actually compare a population. Now, by the time you compare a population of the whole genome of a particular population together, right? You could actually find a conserved region. Now, this conserved region is a particular region of fragmented DNA sequences that is similar in all of the DNA of the genome or DNA sequence of gene. Is that clear? Is that clear? What's that? Oh, thank you. Thank you. Are you guys getting my point? Sorry, it seems I'm fast. Sorry for that. So what I'm just trying to say in essence is this. All right, what I'm trying to say in essence is this. When you compare organisms that is in a population, you could find conserved regions. These conserved regions are regions of similarity that is found in all the population or some of the population. That's what I was talking about, copy number of genes based on the first questions that I was answering. You get a point now? Because by the time you compare two organisms of different species together, you could find conserved region. And this conserved region shows that these two organisms of different species are identical at this region. So if there's a particular gene at that region, it means that the gene that is functioning for a particular structure in that particular organism is also doing the same in the other organism. Is that clear now? So now, how can we make phylogeny between two different species? All right. So that would be covered. So the phylogeny here means phylogenetic analysis such that you'll be able to cover the evolutionary. You want to understand evolutionary differences between these two stuff. So we have which software would you use? Free, of course. Okay. Which software should I don't understand this, sorry. So okay, free, like free softwares. So we have free pipeline tools actually that you guys can work with. Then when you talk about comparative genomics, you often mention, okay, I think we've answered this. I hope I answered that. Please, if I answered that, you can make statements, okay? How can we use transcriptomics to compare the gene expression between two organisms? So for you to use transcriptomics is just that you need to get sequences from two different samples compared to a particular strain, and you call for gene expression, and then you compare these expressions to know those genes that are expressed and those that are knocked down. All right, those are upregulated and those that are downregulated. So that's how you know. Now, how can one apply comparative genomics in metagenomic study for samples or data set from a lake? Yeah, you can actually do that. So let's just go through the practical. Probably we'll cover some of these things, questions you guys are asking. Then as conserved region, you are talking about ITS region. So yeah, even ITS regions, we could have some kind of genetic mutation, because when you're talking about ITS regions, you can actually work with ITS like 16S RNA sequences like short transcriptomics ITS like interspace transcript spacer. So by the time you compare, you could have it could be ITS of the same 16S RNA from different species, or the same species, different strains. But by the time you compare, you could see variations, because this ITS is used to study evolution, all right, between these two sequences. I hope this is very, very clear. I've answered your questions. I think we have another question here on Microsoft Team. In eukaryotic, only regions are why are introns more than exons? So that could be probably to balance the genetic structure, to put these things in order, because not all introns, not all the DNA sequences are actually coding for an amino acid in particular, based on the expressions of those genes in the gene. Then we have what is the duration of this workshop? Okay, so we're going to, it's actually like 2 hours, all right? So we'll end before 3 p.m. GMT. So how will certificate be given? So please, tomorrow we'll talk about certificate for all the participants that have joined us today. So make sure you join us tomorrow to complete the hands-on practical workshop. Recording also will be provided, so all the details concerning the activation key of the Genome software and what's it called right now? The activation key of the Genome software and the certificate, all right, for the workshop will also be shared tomorrow. We have another question here. What is the significance of non-coding regions and DNA sequence? Like I said, some of these non-coding regions are there just to put some balance. Like you don't say that they are not functioning there, because by the time you remove them at the genetic level, they could have effects. They could affect the frame shift and assault. If there's a frame shift at the genetic level, oh, there could be serious alterations. All right, there could be serious alterations or modifications at the protein level. So they are there probably to put some balance in order, to put some order, so such that by the time they get to the RNA, before the mRNA, there could be some splicing. Okay, I hope that answered your questions. So thank you guys for these questions. Thank you for these questions. Now we're going to proceed to the next to the practical sessions. Okay. So I've sent the link. Don't forget to follow us on LinkedIn. All right. I've sent a link for you to download the Genome software, which is the software I'm going to be using for some kind of comparative analysis in this particular workshop. All right. In this particular workshop. All right. So Yamne Puda, what is, okay, are you saying it's, do elaborate it? So are you still talking about the ITS? No, we are not having any Zoom link, please. The link is taken to Zoom. We don't have any Zoom link right now, okay? So please, probably we answer your questions later. Let's just dive into the practical because we don't have much time. Yeah, exon, yeah. Okay, exon and intron. Okay, you want me to answer that? Okay, exon, yeah. So exons are what actually code for a particular protein, functional protein. All right, exon code for a functional genes or proteins. All right, those are exons. They are the open reading frames that code for a functional CDS, which is coding sequence. All right, those are exons, but introns does not code for anything. They're just there to keep the balance. They don't code for anything, but it is actually the exons that code for functional proteins, which is the CDS coding sequences. All right. Okay, So you guys are asking some kind of questions that is not involved in this workshop, but just enjoy this time with us, okay? Enjoy the time with us. So let's proceed to the practical sessions, guys. Let's proceed to the class. So the first practical session we have here is what? Data retrieval from what? From the database. So wherever we stop today, we're going to complete it tomorrow, okay? Wherever we stop today, we're going to complete tomorrow. So just go to NCBI database. I believe you can see my screen. Yeah. So now go to NCBI database. Go to your, go to your browser and search for NCBI database. All right, NCBI database. Now, NCBI database is an online storage. All right, where genomes has been stored, genes are being stored, informations on genetic materials are being stored, some research papers on mathematics are being stored, and the likes. So, and then on NCBI, we can actually do some kind of online data analysis as well, like blasting, which is part of the comparative genomics as well. All right, blasting. So, but this section is much more advanced than blasting. So we need to look at the basic to advanced stuff of what we are here to engage. Okay, so this is NCBI database. I hope you can see my screen, guys. Right? I hope you can see my screen. Okay, so let's proceed. All right. So this is NCBI database, and in this database, we're going to be covering a lot, and we're going to be covering data retrieval. Now, there's something called next generational sequencing, which is NGS whole genome sequencing. So because of our time, we're not going to work with animal data or plant data, but the concept is the same, okay? I repeat myself. We're not going to get animal data or plant data. The concept is the same. So you can follow this concept and work on plant data, whole genome data, all right, on plant whole genome data. Now, for us to proceed in data retrieval, I will talk to you about all databases. Now, this is all databases on on databases, which give us specific areas where different kind of data are being stored. We have the assembly, we have the genome, we have the gene, and then we have the nucleotide. All right, we have the nucleotide. So we can actually search for a particular genome of a bacteria, all right, from this end by clicking genome, and then we can look for Staphylococcus, or let's work with E. coli. Between the genome of E. coli and Staphylococcus, which one is okay? I think let's work with Staph. Right. So for you to get a genome, you could just say WGS Staphylococcus. So we're going to work with it. This is just at genus level. So let's see what we have. WGS here means whole genome, but let's probably remove this and search directly like this. So it's still loading. All right. All right. So as you can see, we have like 75 of it. All right, 75. Now there's another way you can actually search. So you can search. Let's go to NCBI database. This is just at genome level, and for you to download this, you just need to. So you can actually click genomes here, and by the time you click genomes, okay, let's click genomes here, Staphylococcus, and let's click genomes. I don't know why my network is kind of slow. So another way you can do this is go to nucleotide and search for complete genome. All right, can you see? Staphylococcus agentius, we have Staphylococcus aureus. So we keep having them because we search at the what? At the genus level. So imagine if we want to search at G level or at the species level. Sorry, yeah, genus level. So let's say we want to search at the species level rather. So let's just click this. All right, so now we have them. Can you see? Complete Staphylococcus aureus complete what? Genome. And as you can see, here are the complete genome. So we can download the complete genome from here. So now, the idea about this workshop is that you need data for you to do comparative genomics, and this data is NGS data. So we want to get the NGS data of bacteria because bacterial genome is small. Because of our time, we can run faster. So you can use the same thing to do for whole genome sequence of plants. I hope that is very clear. So we are retrieving the old annotated, whole genome sequence of this particular Staphylococcus aureus, all right, whole genome sequence. And by the time you scroll down, you see quite a lot of complete genome. Now you see like 2 million base pairs. Now this is the complete genome of Staphylococcus what? Aureus. Complete genome, complete chromosome of Staphylococcus what? Aureus. Now you can download as many as possible because each of these are strains. This is a strain. This is a strain. This is a strain. All right. These are strains. There are a lot of them. There are a lot of strains of Staphylococcus aureus here, samples here. So let's see what's this giving us. So as you can see, we click the genome from the other hand. All right, from the first way. So it's giving us a kind of like 88,488 genomes. So you could download them from here, and you could download also from here. So you could use nucleotide method, you could use the genome method, all right, to download sequences. Now, remember we're doing comparative genomics, so it means we have to work with different numbers of genomes. You understand? Different numbers of genomes for comparative genomics, because it is when you compare your own genome with as many genomes as possible before you can actually come up with a confident result and report claiming your conserved genes, specific genes of interest. I hope that is very, very clear, guys. I need a reply to the chat room. The data retrieval process, I hope you guys are getting it. Quickly at the chat room. Is it clear? Reply with a yes or no. Wow, screen is not showing. Are you sure? All right, can you see my screen? Can you all see my screen? Hello? Yeah, can you see my screen? Can you see it now? I hope you can see it now. Before we proceed, okay? Can you see my screen? All right, I need more reply. Can you see my screen? Guys, I need reply at the chat room at the Microsoft Teams. Is my screen seen? Please, I need reply. All right, thank you. Thank you. So let's proceed. All right. Now, so I was saying previously, two methods of retrieving the whole genome sequence of an organism. So we search from here. If you don't know how again anymore, you could just like work with, you know, you could like work with the samples. I mean, the video when you get it. Okay. So this is a way of getting the genomes, the whole genome sequence of this Staphylococcus aureus from NCBI database. So just click this line. So, and then go to downloads. So from this download, you could see like genome sequences. So now remember, we actually need the annotated features. So you could like click the annotated sequence annotation. So we could get the sequence and annotation. All right, sequence and annotation. So instead of sequence annotation, so we could get it complete at it. So that this will be the gene bank of this particular sample. Now remember, what I'm about to download right now is the reference sample of Staphylococcus agnatis. All right, this is Staphylococcus agnatis, not aureus now, agnatis. So I could download right now. So you could even do that for Staphylococcus aureus, okay? And then click this. So just get the this one, gene bank only or reference sec only or all. So you could like sequence and annotation, and then when you scroll down, you'll be able to download it. You could save the file into something else. So let's download. So it's downloading. It's downloading right now. So while that is downloading, so how do you download for this method as well? So you just need to click here right now. Okay, you need to click here. Scroll back up, click send to. All right, click send to. Click send to, and complete record, click file. Format should be gene bank full. All right, gene bank full, and then you create. So whichever one you used. So let's work with Staphylococcus aureus because we've started with Staphylococcus aureus already. But I already told you about this. So, but let's work with this method right now. Okay, let's work with this method right now. So let me see the downloads. What do we have? So I think it's still downloading. Okay, so now let's check the results that we have. Yeah, so this is the one with the genome option. This is the one with the nucleotide option. So this one is the one we download like this from this end with the nucleotide option. Okay. So now let me open it so that you see the DNA sequence of this organism. We downloaded the gene bank. All right. So now this is the gene bank. Can you see? We've downloaded the sequence with the gene bank with the sequence coding for each gene. Now this is how it looks like. All right, the sequence gene bank. All right. So this is it, and we could download others as well. All right, we could download others as well. Staphylococcus aureus complete what genome. So now we've downloaded the first one, so we can download as many as possible and extract it. So let's do it this way. Do it this way. Do it this way. All right. But you can choose to download it one after the other if you want. File, gene bank, and create file. Create file means you are downloading. So let's open this file and try to rename this file. Let's try to rename this file. Sorry, my system is a bit slow. All right, so let's rename this. So this with a string. All right, so I'm renaming this. Remove this. Okay, let's correct this. So you could just use the string name, the code alone to save it, so that you can identify it while saving. All right. So the same with this. You have what staphylococcus areas strain. This. So this is the strain. This is the strain. So let's rename it. Done. All right. So this is how you do for all the for each of those genomes. So while you are doing comparative genomics, you could like work with 10 samples, 15 samples. You could work with 20 samples, depending on whichever one you want to work with. So just download them in this way. So we've downloaded 3. Okay, it's not responding. Let's wait. All right, so this would be the 4th one. All right, this would be the 4th one. Different strains. All right, so let's save this other one. This is the strain. So this is the 1st one, right? 4th one. All right. So where's the strain? This is the strain ID. All right. And let's paste it. So we have 4 of it. So let me just try to refresh. So this is 1, 2, 3, 4. Now we downloaded 4 genomes of Staphylococcus aureus, but listen, this is very important. This approach is similar to plants and animal genomes. I repeat myself. This approach is similar to animal and okay, the screen is freezing. I hope it's not freezing now. Is it still freezing? I hope it's no longer freezing. Guys, I hope it's no longer freezing. I need a reply. Yeah, still freezed. Are you sure? All right, let me stop the camera. Okay. What about now? You will get a recording today, okay? What about now? Is it still freezed? Okay, let me stop sharing so that I can share back. All right, I'm sharing back. Select 4. Good. I hope you can see my screen now. Okay, now it's perfect. All right, so let's proceed. So, like I said, now we have 4 genomes. So in case you're working with plant samples or any other kind of sample, this is how you get the genomes. So probably you are doing a research and you get a sample, you have to do genome assembly and genome annotation stuff of the team. Do you get the point now? To get this kind of whole genome sequence and then the annotated one. So which is why probably tomorrow I'm going to show you where you can do some kind of bacterial annotation, okay? But this is for Staphylococcus aureus, and we got 4. Now, out of these 4, one has to be your reference sequence whenever you want to do comparative genome analysis. Whenever you want to do comparative genome analysis. So, but this is how you get data sample. This is how you treat data from whole genome, from NCBI database. So I believe this aspect of data retrieval, I believe this aspect of data retrieval is solved from the database. So the next thing we're going to now is some software tools for comparative genomics. Software tools for comparative genomics. So the first software tools we're going to be exploring today is the Genome software, which we've sent the link to you on WhatsApp. All right, we've sent the link to you on WhatsApp. This is Genome software. That by the time you install it, all right, by the time you install it, you will see something like this. You'll probably see something like this. By the time you install it, probably this could be kind of longer. I could see something like this. So you could just cancel this so that you can have a full detailed overview. Now, this is Genome software. This is Genome software. By tomorrow, we're going to show you how to activate it. But for now, please, we're asking you to use the trial version, okay? Use the trial version. Now, the moment you're done with the trial version, if you're before you install, okay, if you have 12 gig RAM, please download 64 bytes for you can't hear me. Please, can you hear me? Hello, can you hear me? So who's not hearing? Is it the Google Meet people? Okay, can you all hear me? Hello, please, can you all hear me? Can they hear me now? Sounds like your mic isn't working. Hmm? Yes, unmute yours. Can you hear me now? It's not gonna work. To unmute on Microsoft Teams. Please, we are coming, okay? Oh, yeah. Hello? Can you all hear me? Hello? All right, at the Microsoft team, can you hear me audibly? Am I audible? Am I audible? Amazing. Thank you guys for replying at Microsoft team. Now at Google Meet, can you guys hear me? All right, amazing. So let's proceed. So I was talking about Genius software. And this is Genius software. All right. So this Genius software costs like $200 per year for students, but we have the activation. So tomorrow, whatever the case may be, tomorrow we're going to talk about that. But please, the moment you install it, activate free trial. Use trial to activate. And the moment you are done, I was trying to talk about the space of your laptop. So if you have 64 bytes, I mean, if you're using 64 bytes, the requirement of your laptop, and then you have high gig memory, so you can have more memory because this particular software work with the memory of a laptop. So the memory of your laptop, the higher the space you have to run analysis, the more faster your analysis works, right? Faster your analysis works. So to proceed from here, by the time you install it, after tools, go to plugins. All right, go to plugins. Make sure you install all plugins. I'm going to install, right? So I can show you how you can install. By tomorrow, installation, right? Just let me know. So now I find it like this. Make sure I'm online installing. Install. So it's going to be installed. Now it's successfully installed. Make sure you install every, and by the time you click OK. Now, the next thing is go to general. Go to now, if you have 1,000 RAM, you can actually, I only have here on this laptop. So that's why I'm like 3,400. If you're having 2 RAM, you cannot go beyond space. The more the space you have, the more bigger the data you can analyze. So because if you're running bigger data, capacity is more than that present. It's going to tell you that it can go on. You get the point. So this is just, and I already showed you example exploring. Let's a DNA sequence. So wrap it. By the time you click the DNA sequence, wrap it. And by the time you wrap it, you get sequences and you translate. So you can view the complementary strand of your DNA. So this is like genetic analysis, the DNA that you upload. Right? So we are looking forward to giving you guys quality because we have time impact, and this is our motto. This is all we are always saying. You guys, people out there being impacted by our works, by what we do. So which is why we're going to give you guys. That's why we give you guys you know, quality, right? Even if it's free, it doesn't matter. So this is the complementary, and this graph is also about the graph of expression of each of these code, all right? The expression of this code. That is it basically, right? That's it basically for the DNA sequence. Dot plot, just comparing sequences together. Look at the DNA fold. You could wait on the fold is done for this particular DNA. So is it a computing fold? Please wait. So once you wait for some time, it's going to actually, we don't have time for that. You can try it on your own. It's the text of DNA sequences, and this is the info. So imagine if uploaded sequence series, right? So we can do right. So we are just going to work with uploading the continuous software. So as such, we will cover, or as such, we are able to cover data retrieval from database for analysis. It's one of the software actually, which is genome software. Tomorrow we are going to work with software to find genome analysis. All right. All right. And then we're really tomorrow's whatever comparative terms. Okay. So and then we'll look at general properties because we're almost out of time, right? So now let's go back to genome software. So we start working with samples after install plugins, which you're ready to use it. The plugins are individual tools that are out separately that you actually use online, some kind of analysis, assembly, expression analysis, mutation stuff, because that's what this particular software can do. Meanwhile, at this fact, we will actually look, work because you install the plugins, right? So now let's go to file. So before you click file, make sure you click local. You want to click local, click file, new folder, click local, file, new folder, right? Now you can rename this folder. You can rename it like comparative genomics of bacteria. Okay, bacteria right now, which is now. Okay, can you see it here? Now it's done. So we need to upload the sample. So to upload the sample, we need to click the file. Go to file, like click this folder that we just clicked. Go to file. Come to import from file. So right now we're going to look on how we import ever we've done that. So we click PC, go to where you uploaded it, and then look for it. All right. We gotta look for it, okay? I think they are all scattered, right? So here are they. And let's import. So we're importing all the sequences at once, and it's processing the documents. It's processing the. So keep sequence separated. This option, keep sequence separated. Keep sequence separated. So it's importing them one after the other, including the name of the file. So, but this actually I think is based on the session. Yeah, it's based on the session number. But it's uploading right now. So we're uploading this whole genome sequence. Already annotated one. So we're uploading Chimba because we downloaded the sequence together with Chimba. That's what first. All right, so now it's completely done. This is the whole genome sequence of the 4. So you can have as many as this, download as many as this. Guys. So just I'm going to give you guys assignments. So we're actually viewing, as you can see here, you can see some kind of a view. So this is viewing all the 4 genomes. But more than 4 genomes may not be able to see this way because it tells you that the document is. You just have to view one after. So now viewing one after the other, you just previous short sequence, right? So now you can see, so you could maximize. Now maximize it, so you could like zoom in. Zooming in here the circular genome of this particular sequence and circular sequence. Now you do it in a linear format. You could wrap it as well, or you could do it in a linear, and then you view zoom it. In zooming, you'll be able to get access to CDS, right? The CDS, the gene, the coding sequences, and everything is present in this particular genome. These are the genomes that we're going to and do so. So now, for you to see the entire genome sequence present, RNA, the tRNA, and tRNA, rRNA. These are the functional proteins. What is each of the CDS, right? And this is just the basic. By tomorrow, I hope we have this tomorrow. So we're going to look at genome properties table, concept of research paper. Maybe whenever you want to come with a research paper. And then we have the genome assembly, comparison, using the genome software analysis. And then we have the particular genome manipulation. I will just do if we got an example to get the kind of genome sequence properties, chromosome properties, just like the CDF stuff. Awesome. Then we have the last assignment. I hope you have okay, because this is quite a lot and we're time. So, but prepare to tomorrow. So tomorrow, no much or theoretical sessions, and then we end the workshop. So right now we've come to the end of this